Our fifth learning unit that we have here is very related to learning unit 4 on electron transport and oxidative phosphorylation in mitochondria, and that's photosynthesis as it occurs in chloroplasts. So you might say, why are we learning about plants? But it's a way to reinforce concepts that we learned about the organelle, the mitochondria, and see homology in a lot of what happens in chloroplasts. So not only homology with some of the same functions, but maybe somehow how some of these things are different. And so a lot of compare and contrast is what we'll do between mitochondria and chloroplasts. And so there's going to be a lot of things that are similar, but then again, a lot of things that are different. And one of the things I hope you take with you is this understanding that plants are pretty amazing, and that during the day they can use chloroplasts as an important organelle to generate ATP and NADPH. But at night, when there's not light available to drive those biochemical reactions in the chloroplast, plants are just like us in the sense that they need to use their stored carbohydrate reserves, which are starch. And again, they break those down into glucose, and then they use their mitochondria to, after we break down glucose and glycolysis, go through the citric acid cycle, electron transport, oxidative phosphorylation, the pentose phosphate pathway. All of that same chemistry that happens in animal cells happens in plant cells at night. So really unique sort of biochemistry, a great way to sort of integrate concepts that we learned in electron transport and oxidative phosphorylation, review them, and then apply them with plants. So we sort of wrap up our discussion sort of thinking about applications with this, and importantly thinking about the importance of having this enzyme rubisco as a mechanism for fixing carbon dioxide. So two gases that are present in our atmosphere, which are relatively inert and thus unavailable to us, are carbon dioxide as a carbon source, and then N2 gas, nitrogen gas, as a nitrogen source. But as we'll learn about when we've learned about in this chapter, the ability to fix carbon in a usable form as carbohydrates. Rubisco is responsible for doing that. We'll learn about the enzyme nitrogenase when we learn about nitrogen in our amino acid chapter. And we'll learn about the importance of organisms able to do that kind of chemistry, to take what essentially is two nitrogen atoms connected via a triple bond. That is an inert gas, and convert it into a bioavailable form by converting it into ammonia or amines. And so really important chemistry happens in plants. We wouldn't be able to survive without it.